A good reason to learn about neurotransmitters is that drugs that work on the brain do their work by affecting neurotransmitters. So when you start talking about drugs, sedative drugs, stimulant drugs, and so on, psychedelic drugs, drugs that change mood, drugs that change behavior, they're causing their effects by working on neurotransmitters. So let's look at a couple of important concepts here. The concept of an agonist and an antagonist. So neurotransmitter effects can be enhanced or inhibited by drugs and toxins. So again, drugs causing effects that we want to cause, toxins usually causing unintentional effects. But they can either enhance a neurotransmitter or they can inhibit a neurotransmitter. So for example, you could have a drug or a toxin that caused a neuron to make more neurotransmitters, or maybe make less neurotransmitter. Or you can have a drug or a toxin that caused extra neurotransmitters to get released, or it might prevent neurotransmitters from getting released. We already saw that when we looked at the neuromuscular junction. Remember, we looked at cobra venom and botulinum toxin, and we looked at taipan toxin. We saw how those things can change the action at a synapse, and that's what various drugs and toxins can do. But they're working on neurotransmitters. That's one of the main ways. And again, remember how nerve gases, for example, blocked acetylcholinesterase so that it couldn't remove acetylcholine from the synapse? Same thing. A drug or a toxin might cause extra neurotransmitters to get removed from the synapse, or it might prevent neurotransmitters from getting removed from the synapse. Either way. And the receptor site itself, we saw that with the cobra. You can block the neurotransmitter, or it can happen that some drugs or toxins can pretend to be the neurotransmitter and actually open the receptor, all right? So let's make sure we know these words. Agonist is anything that enhances the neurotransmitter. It causes more of an effect of that neurotransmitter. That's what the word agonist means. The word antagonist, A-N-T, against, is anything that inhibits the action of a neurotransmitter. All right? So again, a lot of drugs and toxins function either as agonists of a neurotransmitter, causing more of that neurotransmitter's effect, or they function as antagonists, causing less of that neurotransmitter's effect. Drugs and toxins function as agonists and antagonists. All right? Now, you will find some sources that want to use the term agonist and antagonist only for things that affect the receptor site, but that's a very strict interpretation of that term. We're going to use it in the broader sense that most textbooks talk about. So if you look on the lower right, you can see their 5-HT receptor, all right? 5-HT3, that's a subtype. 5-HT is serotonin. So notice how there looks like a little, puzzle piece that says 5-HT, that little blue puzzle piece. See, that would normally fit into the channel, and that would fit into the receptor, open the channel, and sodium can flow through. Look, though, over on the right panel of that illustration, you can see that yellow square. It's an antagonist. That would be a drug or a toxin that's blocking the binding site for 5-HT, 5-HT being another name for serotonin, and therefore that's preventing the channel from opening. That would be an antagonist. It prevents the channel from opening. And in the upper right, you can see antagonist drugs can decrease the release of neurotransmitters, so there's fewer, all right? They can also help destroy neurotransmitters in the synapse. They can also mimic neurotransmitters. Lots of different ways to be an agonist or an antagonist. So here, oftentimes they use the analogy of a lock and key mechanism. So you can think of as an agonist as a key that unlocks the lock. It opens the lock. Whereas an antagonist is something that prevents the lock from opening. So take the example of acetylcholine. There you can see in the upper left, that's an acetylcholine molecule. Notice it has that purple part and the blue part. Then notice right below it is carbachol, which is an agonist. It will unlock the acetylcholine receptor. Then in the very bottom is tubocurarine, which is an antagonist. That will block the receptor. So you see, that's what's going on. molecules that are not exactly the neurotransmitter can in some ways pretend to be the neurotransmitter. They can pretend to be the neurotransmitter in a way that opens the channel just like the neurotransmitter does, or they can pretend to be the neurotransmitter in a way that blocks the opening of the channel, all right? So you can think of an antagonist as something like partially fits the lock but doesn't open it. You know, think about it. A lot of house keys look the same. Right? My kids, they live in houses. My house, the house keys look the same. But if I stick my house key into the lock on their house, it'll fit into the lock, but it won't turn the lock. That would be like an antagonist. Fits the lock, but doesn't actually turn the lock. So carbachol is an agonist, all right? Tubocurarine is an antagonist. So which portion of the ACH molecule binds to the receptor and opens it? So notice there's the purple part and the blue part. Noting that carbachol is an agonist and tubocurarine is an antagonist, looks like maybe one of those two, of the purple part and the blue part, one of those might be what actually opens the channel, whereas the other one doesn't open it. So which part opens it? Yeah, the purple part. See, carbachol has the purple part, so that actually opens the channel. Whereas tubocurarine has the blue part, which would be like the lock, the key fitting into the lock that it won't turn. So it'll partly fit into the acetylcholine lock, but it can't turn the lock, okay, because it doesn't have that purple part. That's the difference between an agonist and an antagonist. Here's that same concept again. Locks work by tumblers. I don't know if you're a professional thief, lockpick, you probably know all about this. You know, you see in the upper half of the illustration, there's a key that will fit into the lock. But you know, the little, all those little cuts on the key, all those little bumps on the key, notice they don't match up correctly with the tumblers, therefore the lock won't turn. But on the bottom, there's a key that when it goes in, all the little bumpies on the key exactly fit the configuration of the tumblers so that they line up correctly, and then the lock opens. So the top is like an antagonist, doesn't open the lock, even though the key fits in. Bottom is an agonist. Not only does the key fit in, it actually opens the lock. So let's practice. Let's take a look at some examples here. So strychnine, component of rat poisons, it blocks postsynaptic glycine receptors. So glycine is a neurotransmitter. So I'm telling you that strychnine, which is found in some rat poisons, not much anymore, but in the old days it was, it blocks the receptor. So would that be an agonist or an antagonist? Are you playing the home version of the game here? What would that be? That would be an antagonist because it blocks the receptor, prevents it from opening. How about Huen toxin? Toxin in the venom of Selenocosmia huena, the Chinese bird spider, which opens calcium channels in the axon terminal, thus increasing the release of neurotransmitter. So these are cool. These are big tarantula-like spiders. They live up in trees, and they're camouflaged. They blend in, and a poor little birdie lands on a branch, and the spider jumps on it, injects it with venom, then it dies and it eats it. It's just a Disney movie, I'm pretty sure. So what would that be? So in this case, it opens the calcium channels. Therefore, it would be an agonist. And you're thinking, well, wait a minute. Why is it a toxin if it's helping the neurotransmitter? Remember homeostasis? Too much of a good thing? See? You can't have too much neurotransmitter. You can't have too little. You have to have just the right amount. So in this case, the toxin causes extra calcium release, and ultimately that causes spastic paralysis. Okay, diisopropyl phosphorofluoridate, DFP, an insecticide that inhibits acetylcholinesterase, ACHE, the enzyme that breaks down acetylcholine in the synapse. So here we have an insecticide, and what it does is it blocks acetylcholinesterase. So is this going to be an agonist or an antagonist? Be careful. It's an agonist. You're thinking, wait, it inhibits. I thought that was an antagonist. No, you have to pay attention. Don't just key on the word block or inhibit. You've got to actually think about what it does. So remember, acetylcholinesterase was the enzyme that takes acetylcholine out of the synapse. So if you inhibit that enzyme, then it means extra acetylcholine stays in the synapse, and therefore we have extra effect of the neurotransmitter. That's an agonist. Notice that works the same way as nerve gas does. These are the organophosphate pesticides, work much the same way nerve gases do. All right, they're agonists, but they're still toxins because, again, too much neurotransmitter is bad. Remember, nerve gas causes spastic paralysis. Okay, muscimol, found in Amanita muscaria mushrooms, it binds to postsynaptic GABA receptor sites. So GABA is a neurotransmitter. So these are not the psychedelic mushrooms, okay? These are actually very poisonous mushrooms. They used to make something called fly agaric, which is a poison that people used against flies back in the old days. You mash up the mushroom. Flies are attracted to it if they eat it, and then they die. So they bind to postsynaptic GABA receptor sites. That's what muscimol does. So agonist or antagonist? Yeah, agonist, because remember, binds to the receptor means it opens the receptor. All right. So muscimol opens the GABA receptor, GABA being a neurotransmitter, and that's an agonist because it causes more of an effect of the neurotransmitter. Okay, reserpine, component of a drug called Diupress, which is an antihypertensive drug. What does that mean, antihypertensive? That's a drug that lowers blood pressure. It inhibits the storage of catecholamines in presynaptic vesicles. So remember, inside the axon terminal, you have vesicles that hold neurotransmitter. And then, when the action potential makes it down to the axon terminal, it causes the release of the neurotransmitter inside of those vesicles. So in this case, the vesicles are going to have less neurotransmitter inside. That's what this drug does. It causes the vesicle to contain less neurotransmitter. So therefore, what's that going to be? That's going to be an antagonist. Because now when exocytosis happens, there won't be as much neurotransmitter as there should be, because the vesicles don't have as much as they should because of the drug. The drug caused there to be less neurotransmitter in the vesicle. See? Got to think these all the way through, but they're always logical, always make sense. Just think in terms of, does it enhance the effect of the neurotransmitter, or does it inhibit it? That'll always get you to the right answer. Okay, so a couple more. Cocaine stimulates the release of norepinephrine and dopamine from presynaptic terminals. So it causes more norepinephrine and dopamine to get released, therefore it's an agonist. Yes, indeed it is. So sertraline, which is sold as Zoloft, fluoxetine sold as Prozac, paroxetine sold as Paxil, these are popular antidepressant drugs. And what they do is they block the reuptake of serotonin. So remember, there are two ways to get neurotransmitters out of a synapse, either enzymatic degradation or reuptake. So these popular antidepressant drugs block the reuptake of serotonin. So what happens? More serotonin stays in the synapse, therefore these drugs are agonists. Again, don't key on the word block. It's that they're blocking the reuptake. Therefore, more neurotransmitter stays in the synapse. Therefore, there's more effect of the neurotransmitter, okay? So, chiriquitoxin, a substance secreted by the skin of Atelopus chiricensis, one of the poison dart frogs of Costa Rica. Have you seen these? They're cool. They're these little brightly colored frogs that live down in the rainforests of Central America. And what they do is they secrete toxins on their skin. And the people who live down there use them as blowgun toxins. They hunt using blowguns. You've seen it, like a long tube, and you have a little dart inside, and you blow it, and then you shoot like at monkeys up in the trees, and the monkey slaps itself where it got shot, and then in moments the monkey's paralyzed and falls to the ground, and then you got monkey for dinner. Dad, what are we having for dinner? Monkey. All right, so, but notice they block voltage-gated sodium channels. So what's that going to do? What did we need voltage-gated sodium channels for? Yeah, we needed those for the action potential. So what happens if you block voltage-gated sodium channels? You get no action potentials. Therefore, these are antagonists. They cause flaccid paralysis. Okay? Dihydroxyphenylserine, an experimental drug converted to norepinephrine in the axon terminals. So if you take this drug, your body will turn this drug into norepinephrine, which is a neurotransmitter. Therefore, what's this drug going to do? Is it going to cause you to have more neurotransmitter or less? Yeah, so you're going to have more because it turns it into neurotransmitter. Okay? On your exam, I'll have a few of these questions like this. I'll give you an example of a drug, explain how it works, and then I'll expect you to tell me whether it's an agonist or antagonist. It's pretty easy. Just look again. Does it enhance the effect of the neurotransmitter, or does it inhibit it? Okay? All right, let's look at our first neurotransmitter here, acetylcholine, which we've seen before. Now let's look at it as a neurotransmitter. So acetylcholine, neurotransmitter at the neuromuscular junction, synthesized from choline and acetic acid. You get those from your diet. Inactivated by acetylcholinesterase. We saw that before. All right. Many neuromuscular disorders involved ACH, remember? So like myasthenia gravis, that was a blocking of acetylcholine receptors, and there are others as well. Many nerve gases and insecticides are ACHE inhibitors. We just talked about that. There you see in the lower right those insecticide cattle ear tags. You've seen those cattle out in the desert? We're open range around here a lot. If you go out in the desert, you see cattle all over the place. They have these ear tags instead of a brand. That's an easy way to mark the cow. Some of them are just tags, but some of them actually have insecticides. So this can be a problem. You know, like at a ranch, you got a box of these lying around, a little toddler gets into them. What do toddlers do with everything they find? They stick it in their mouth. Get a little kid chewing on one of these things, and next thing you know, you got a little kid going into spastic paralysis, all right? Two kinds of receptors, nicotinic, which is ionotropic, and muscarinic. We just saw those a little bit ago. We're not going to really focus on those or emphasize those until we get to the fourth part of the class. When we talk about the autonomic nervous system, those are huge. So I'm just pointing them out now. You don't really need to know much about them for this exam, but on the fourth exam we'll be talking about those a lot. Okay? Look at acetylcholine. It's a little tiny molecule. Not a big deal. Both huge, nicotinic, muscarinic, in the ANS. And it's all fun and games till someone drinks the beer with cigarette butts in it. Yeah, you know, like at parties, you know, people get drunk. You got smokers, and they're like putting their cigarette butts out in old beer cans. And sometimes, you know, somebody can't find their beer can, and they grab one. They think it's theirs, and it turns out it's a beer can that, you know, 14 people put their cigarettes out in. The nicotine leaches out into the water. You can actually get nicotine poisoning. That actually happens. So watch out. I always carry a Sharpie in my pocket, and I mark my beer so that other people won't put their cigarette butts in. Of course they do anyway because they're drunk. So, okay, let's look at some excitatory amino acids. So our first general category of neurotransmitters are amino acid neurotransmitters. So remember, amino acids, these are what make proteins. 20 to 22 amino acids common in the human body. They make the proteins that you use all through your body, both for structure and for function. But some amino acids can double as neurotransmitters. Your body, very efficient, use the same molecule for different purposes. Make proteins out of it, also use it as an amino acid. So amino acid neurotransmitters, these are small molecules composed of an amine, or an amino group, really it should say, NH2, and a carboxyl group, a carboxylic acid, a COOH. All right? So glutamate, our first one, this is an excitatory amino acid neurotransmitter. So what does that mean? What's true about glutamate? What kind of ion channel does glutamate always open? Yeah, it always opens sodium channels. It is the principal excitatory neurotransmitter in the human brain. It's present at about 90% of all the synapses in your brain. Most of the synapses in your brain, you're just trying to get a signal from one neuron to the next neuron. And so you just want that signal to happen as rapidly as you can. And so glutamate is the perfect neurotransmitter because it opens sodium channels, which are fast ionotropic channels, and basically the signal then just essentially jumps from one neuron to the next neuron, all right? Different parts of the brain are sending signals to other parts of the brain. You want the signal to get there as rapidly as possible, okay? There's several types of receptors. This is, again, remember, it's the receptor that matters as much, if not more, than the neurotransmitter itself. So there are AMPA receptors and NMDA receptors. They play a big role in memory and learning. NMDA is N-methyl D-aspartate, and AMPA, I forget all the things in there. It's 3-alpha, 5-methyl, I can't remember. You don't have to know either. But these are subtypes of glutamate receptors. They play a big role again in memory and learning, and also in just simply from moving the signal from one neuron to the next. They are purely excitatory, all right? So what happens if you overexcite these receptors? Well, like you might guess, you end up with things like stroke, autism, epileptic seizures. In other words, you overexcite the brain. If you overexcite an excitatory neurotransmitter, you overexcite the brain itself. And those are the kinds of things that can happen, all right? Again, so important to know neurotransmitters in terms of knowing brain function. Knowing something about the neurotransmitter helps you understand what would happen if you change that neurotransmitter. So the next one, the excitatory amino acid, is aspartate. So another excitatory neurotransmitter also activates the N-methyl-D aspartate receptors. And aspartame, you've heard of aspartame before. That's NutraSweet, they say all that is. That's simply, they discovered it by accident, as I recall. They're just screwing around in the lab. You've taken aspartate, an amino acid, and you glue it onto a phenylalanine, and by golly, it tastes sweet. So they used it as an artificial sweetener. That's why some people say, and I'm actually one of them, that when I drink products with NutraSweet, I get headaches. Nobody really knows why, and people dispute whether that really happens. But it does happen to me. I swear it does. And think about it. When you use NutraSweet, you're basically adding excitatory neurotransmitter to your brain. Is that really what causes the effect? I don't know. But I get headaches if I consume NutraSweet products. Both of these, by the way, glutamate and aspartate, are inactivated by glial cell uptake. So it's glial cells that suck the neurotransmitters back in. All right, that's how we get rid of it. So there you go. We've looked at 3 neurotransmitters already. Acetylcholine, which by the way is its own category, it's a quaternary amine. And then we have our first 2 amino acids. They're both excitatory, glutamate and aspartate. Let's continue the cavalcade of transmitters. Oh, I talked over the transition, so that got garbled. I was just saying let's move on. Here we have more amino acids, in this case gamma-aminobutyric acid, which you don't have to know that, just remember it as GABA, G-A-B-A, gamma-aminobutyric acid, GABA. This is the principal inhibitory. So remember, glutamate was the principal excitatory neurotransmitter. This is the principal inhibitory neurotransmitter in the human brain. You have to have both. Remember, homeostasis, balance. You want some neurotransmitters that depolarize. You want some neurotransmitters that hyperpolarize. You've got to have some of both in order to have the right balance in the brain. It's made from glutamate. That's a cute trick. Your body's never going to run out. You make glutamate, and then, if you need some GABA, you just convert the glutamate into GABA. Drugs that function as agonists of GABA are generally known as depressants because, again, these are inhibitory, so they slow the brain down. And main drugs in this category are tranquilizers and sedatives. That's what they're doing. Like there you see in the illustration, Xanax. Xanax. It's an agonist of GABA. So when you take Xanax, it activates lots of GABA receptors. GABA opens chloride potassium channels. It hyperpolarizes membranes. Therefore, it slows the brain down. These are the tranquilizers and the sedatives, okay? They include the benzodiazepines, so the nurses all call them the benzos, such as lorazepam, Ativan, diazepam, Valium, alprazolam, Xanax. You may know they're both generic and trade names for drugs. Nurses will joke about how, some nights they wish they had like an Ativan mister that they could turn on in the ward. You got a bunch of crazy patients, just turn on the Ativan mister and suddenly all the crazies are sleepy and they're calm and so on. So there are lots of others. They're non-benzo, like Ambien, all right? That's one that they use as a sleeping pill. So these also include more potent drugs for seizures and anesthesia, such as propofol, Diprivan, phenytoin, Dilantin, and thiopental, which everybody calls sodium pentothal. These are much more powerful agonists of GABA, and they use them to control seizures. Somebody who's seizing, they'll give them one of these drugs, or they'll use them for anesthesia. All right, propofol, that's what killed Michael Jackson. He was using it recreationally, and took so much that he completely shut his brain down. That's what will happen. Phenytoin, actually a lot of times they'll do that after somebody has brain surgery. Sometimes when you do brain surgery, it increases the chance of seizures, and so they'll just give that as a prophylactic to try to make sure that you don't have seizures. All right. Alcohol is another one, and alcohol is a depressant. That's what alcohol does. It slows your brain down. Look what happens to people who are drinking alcohol. People who are drinking alcohol, they start talking slower. That's why you don't drive, because your reflexes are slower. You see something, you try to hit the brake, you're like 3 times slower than you would be if you didn't have the alcohol. Yeah, it's the bad stuff. So, and in the old days, alcohol was actually used as an anesthetic and as a tranquilizer. I mean, people still do that. Kind of use it as a sleep aid, Have a glass of wine before bed. It may help you sleep better. Not too much. Too much alcohol actually impairs sleep, but a little bit might actually help. Reuptake is another way you get rid of GABA. All right, is the way you do get rid of GABA. Okay? So that was GABA. That was an inhibitory. Next inhibitory, that is glycine. So once more, an inhibitory neurotransmitter. So strychnine, we mentioned that a little bit ago, age-old poison, is a glycine antagonist. So in other words, glycine is an inhibitory neurotransmitter. Strychnine inhibits the inhibitory neurotransmitter, therefore it leads to excitation. So what do you suppose would happen then if you poison someone with strychnine? Yeah, you get convulsions. In other words, if you block an inhibitory neurotransmitter, it would be the same as sort of activating an excitatory neurotransmitter. You would overexcite the brain. And that's how it killed rats, basically. It caused rats to go into convulsions. It used to be common. You go down to the hardware store and buy a box of strychnine. You can see there in the lower half of the illustration. Those are actual labels. They stopped doing that because strychnine turned out to be a favorite drug that people used to kill their wives and husbands. And it was fast acting, and there wasn't really a way to neutralize that at the hospital, so they stopped selling it as a rat poison. Now they use warfarin, which is an anticoagulant. Causes rats to hemorrhage to death from the inside. And the reason they use that now is because they do have good antidotes for that. So if you try to poison your spouse with rat poison now, they'll catch you, you son of a bitch. They'll save your spouse, keep him alive, and you'll be doing 25 to life at San Quentin, making big rocks smaller, okay? Reuptake, the way you get rid of glycine. So those were our 4 amino acid, 2 excitatory, remember, glutamate, aspartate, 2 inhibitory, GABA and glycine. We also had acetylcholine. We've done 5 neurotransmitters so far. Talked over the transition. We're now going to move on to the next category, what we call monoamine neurotransmitters. So monoamines are tiny molecules that are similar to amino acids. They're not exactly. They still have an amino group, okay? And we have categories now for the monoamines, and we're going to get into these more in the next PowerPoint. But you do have to know these categories. So catecholamines, these are a big deal. They're called catecholamines because they all have a catechol group in their chemical structure. So see the catechol over there on the right? It's a benzene ring with a couple of hydroxyl groups. That's a catechol, all right? All of these neurotransmitters that are catecholamines have a catechol group. The catecholamines are norepinephrine, NE we abbreviated, epinephrine, and dopamine, DA. These are big deal neurotransmitters. We talk about these all through the 4th section of the class, and we talk about them a ton in Bio 202. So get your catecholamines down now. First part of Bio 202 is all the cardiovascular system, and these are incredibly important neurotransmitters in the cardiovascular system. So, man, these 3, norepinephrine, epinephrine, dopamine, get to know these backwards, forwards, and inside out. These are not going away for like the next 2 semesters, okay? Then there are indolamines. They have an indole group. So see in the upper right that indole group. So again, just it's a chemical grouping, but it seems to have an effect on what these neurotransmitters do. All the catecholamines have similar functions. They have a catechol group. All the indolamines likewise seem to do similar kinds of things. They have this indole group. The main indolamines are serotonin, which we abbreviate 5-HT. 5-HT stands for 5-hydroxytryptamine. Serotonin is the nickname that you'll see mostly in hospitals and stuff, but like in research, they tend to call it 5-HT. That's its more proper name. And melatonin. Melatonin is very similar to serotonin. Then another monoamine that's neither a catecholamine nor an indolamine is histamine. And you've heard of histamine before, I'm sure, because you've taken drugs that block histamine, haven't you? Think about what those drugs are.